The cardiac muscle cells in the myocardium require activation or stimulation by the intrinsic conduction system in order to contract. Therefore, the intrinsic conduction system will ultimately control heart rate. Atrial cells will depolarize first when stimulated by the sinoatrial node. After a short delay, which is about 100 milliseconds, that corresponds to 0.1 seconds or a tenth of a second, the ventricle cells will depolarize when they are stimulated by the atrioventricular node, the atrioventricular bundle, the right and left bundle branches, and the Purkinje fibers. This delay is going to permit the atria to fully contract before the ventricles begin their contraction. When cardiac muscle is stimulated, either in the atria or in the ventricles, the muscle cells will first depolarize. They will then experience a plateau phase, and then they will repolarize. When cardiac muscle is stimulated, voltage-gated sodium channels will open, creating the depolarization. So here, at this point, is when sodium will enter the cardiac muscle cell. We see that voltage inside of the cell will climb from a resting membrane voltage of either minus 80 or minus 90 millivolts. It will climb up to a maximum of about plus 30 millivolts. Anytime the cell membrane voltage rises above 0 millivolts, this is what we refer to as a depolarization. After that, we see calcium channels will open, and this will allow calcium to trickle into the cell for about 150 milliseconds or so. And it's this calcium that creates the plateau phase of depolarization, where the cell remains activated during this time, generating prolonged contractions that are necessary to pump blood. We also see that calcium allows the myosin proteins in the cardiac muscle cells to bind to actin proteins and pull, just like in skeletal muscle cells. And it's ultimately this that generates the contraction of the muscle. So finally, voltage-gated potassium channels will open, which are going to create the repolarization. Anytime we have voltage decline or drop from a positive to a negative, we call that a repolarization. And this repolarization involves potassium exiting the cardiac muscle cell. We see that the membrane voltage returns to rest, and the muscle cell can now be stimulated again for subsequent contractions and heartbeats. So figure 1921 shows the opening of the voltage-gated sodium channels in the cardiac muscle cell. The tracing over time shows the voltage rising from minus 90 millivolts to plus 30 millivolts. So that's the depolarization. And it's at this point when the voltage rises to plus 30 millivolts, that's when the sodium channels will begin to close. Opening of calcium channels allows calcium to slowly trickle into the cell. Calcium, being positively charged, will prolong the depolarization, creating the plateau phase. And it is only during the depolarization and plateau phase that the cardiac muscle will contract and will continue contracting. So after approximately 150 milliseconds of being open, the calcium channels will close and potassium channels will open. Potassium will exit the cell, and potassium, being positively charged, will cause the membrane voltage to move toward the negative side, back towards negative 90 millivolts, which is the resting state of the muscle cell. By comparison, skeletal muscle action potentials are quite short, creating brief contractions. Of course, if skeletal muscle cells are stimulated rapidly in succession, the muscle will continue to contract until it fatigues. However, that's not shown in this figure. The only thing that we see is a single stimulus and a single contraction. The prolonged action potentials of cardiac muscle cells, on the other hand, create prolonged contraction times, which is quite critical for being able to pump blood effectively through the heart.